Hello, everyone. Today is May 19th, 2020, and it is our first virtual meeting. And we are just so excited to have Jasmine HAIN as our speaker tonight. She is involved in a graduate master's program in biological science at California State University, East Bay. She has been involved with this project with Brian Perry in the lab for the past 4 years. And Jasmine is currently researching an endophytic fungal population of California natural coastal plants, which are, their common name is periwinkle. And she is a graduate here. She's a graduate here. Um, she got her scholarship from us last year in 2019. So without further ado, here is Jasmine HAIN. Hi.
Alrighty. Hello everybody. I'm happy to share my research with you all. I hope you can see me and hear me and everything. Sort of strange new format that we all find ourselves in. So I'm going to be showing you a presentation of my graduate research, which I've been conducting under the lab of Dr. Brian Perry at Cal State East Bay. It's work that I've been plugging away at since 2016 in my undergraduate and sort of carried on over into my master's research. So without further ado, let's go on ahead and get the presentation going. Okay. So my research topic of interest involves documenting and characterizing communities of foliar fungal endophytes in a common coastal succulent. So if any of you have taken a hike along any part of our San Francisco Bay regional shoreline, or even further, you've probably encountered pickleweed, which is imaged here on the front of the presentation. It's pretty innocuous. It's an edible plant. We'll talk a little bit more about it later, but our native species, Salicornia pacifica, has not had any sort of work done to characterize its foliar fungal endophytes. And so really this research sort of raises the questions of, are there any fungal endophytes found in the photosynthetic tissues, so only the above ground tissues? And if so, what do those communities look like? And do those communities change in character based on specific characteristics of the habitat itself? All right, so as I just mentioned, the research investigates the diversity and abundance of foliar endophytes. So we want to look at, how many segments of plant tissue contain fungal endophytes. We sort of started out with that preliminary research, and now it's going to go into some modified methods. So we'll talk about that soon. No such study has yet been conducted on Salicornia pacifica, and given the character of endophytes, which again we will talk about shortly, the environment plays a major factor in the types of fungi that you will find inside of these tissues. So, each coastal ecosystem may have, variations in those fungal endophyte populations. These are annual succulents, they are halophytes, and they are very, very widespread along the west coast of North America. And you can, you find them pretty much everywhere that there is water. All right, so let's get a little bit into foliar endophytic fungi, a little bit different from some of the more interesting charismatic fungi that you may find out collecting. Those are absolutely lovely, but I decided to focus on something a little less visual. So they are primarily ascomycetes, and they are found in photosynthetic tissues. And you see I've given you a little diagram here. And really what makes them distinct from plant pathogens, and even though those lines, I will go into this a little bit later, those lines are not quite as clear as we thought they were. They live inside of plant tissues asymptomatically, which means that they don't cause any sort of damage to the plant itself. And in fact, there's a lot of evidence that suggests that they may actually assist the plant with specific functions, and in relation to this research, may assist the plants with actually enduring these what we would consider to be harsh environments. So the halophilic character of Salicornia pacifica may have so much to do with the physical sort of composition of the plant and with its microbiota, which would of course include foliar fungal endophytes. So fungal endophytes are found consistently in land plants. I'm not aware of any cases where there were not fungal endophytes present. So this is sort of a burgeoning area of research. Maybe not burgeoning because there's been quite a bit of research on it over the past 30 or 40 years, but really going out and documenting the breadth and variety of different fungal endophytes and what types are found in what ecosystems really is work that needs to be done. And so we're trying to add a little drop to that bucket. And so, they play a really important role. There's a lot of really interesting research coming out that suggests that they have an influence on herbivory. So they may or may not influence the, sort of susceptibility of a plant to being eaten by an insect. And then nutrient uptake is another big one. So endophytic fungi and bacteria have been found to assist with certain metabolic processes as well as just uptake of nutrients. So super, super cool little critters, this sort of secret world of microscopic fungi that we sort of write off as, black molds or dark molds. But there's actually, there's a breadth of interesting information there that we haven't necessarily explored in its entirety. All right, so within endophytic fungi, and this is sort of a precursor to some of the hypotheses and statements that I'll go into a little bit later with this research. There are two distinct lineages that we've been able to identify, and this, of course, is still, in a state of dynamic change. But it has been widely sort of considered that there are two major lineages of endophytic fungi. So they're claviciptaceous and non-claviciptaceous. And this really just is a distinction between grass-associated endophytes and everything else. So sort of interesting that there are unique lineages for grasses and non-grass plants. And of course, we're working with a succulent angiosperm, all of that. And, amongst these non-claviciptaceous endophytes, we can actually identify a couple more lineages, which again are sort of in a state of adjustment and flux, and we're changing these definitions all the time. But, currently we sort of understand that there are these specific classes of endophytes, and class 2 endophytes are really, what we would be expecting to see in this study. So these endophytes are known to be associated with a wide diversity of plants, and they're also known to exist in plants that have an affinity for extreme environments. So what makes them really unique is that they can both be sort of transmitted and move from one plant to another via the spores that they produce. So when the plant dies naturally, the fungus will take over and start to digest the plant. It sort of uses its endophytic mutualism to get to that food, food source first. But there are, so we can assume that there are spore rain. We can assume that there is wind dispersal of those spores, which we're all very familiar with. But there is also evidence that some of these endophytes may actually be vertically transmitted, which essentially means that they may be present in the seeds of those plants. And so they may be transmitted from generation to generation, which is something that is more commonly seen in grasses. So it's sort of a unique, character of this specific class of non-claviciptaceous endophytes. And that's going to sort of come into play a little bit later. So a lot of the research that exists involves bacterial endophytes, and much of it involves arbuscular mycorrhizae, which are those fungi that are associated with plant roots. And those are very, very important and super cool. But again, there is not a ton of information about those foliar endophyte systems in communities. We do know that root bacterial microbiota, they contribute to tolerance to salinity for certain species of plants that live in close association with members of the genus Salicornia. And those bacteria, and there's some fungi as well, they actually upregulate genes that sort of manage being in that high salt environment. So we know that there is some sort of exchange between the plant and these sort of rhizosphere soil associated fungi and bacteria. But again, we don't know a ton about what's going on in the phyllosphere. So something that is, it sort of was difficult for me to wrap my head around and ended up providing, some interesting sort of framework for my results that I obtained in preliminary study. Fungal endophytes actually have a range of expression. So what we're finding as we go out and we sample more plants and we sort of gather more information, we're finding that fungal endophytes may be a pathogen in one plant and then a endophytic mutualist or commensal in another. So that is totally, unique and interesting. And it poses a lot of questions, right? So you can have some pretty deadly plant pathogens to a certain group of plants existing in another plant that, the plant looks totally fine. It looks totally healthy. And so there's definitely some sort of connection and relation between the plant's natural biological response to sort of harboring and taking in these spores, and once they germinate inside of the plant tissues, there is some sort of relationship and some sort of host selection that's going on here, which is pretty interesting. And there's been a lot of research done on Spartina, so cord grasses. Those have been studied somewhat more so, at least, than Salicornia, and we've gathered some really interesting insights. All right. So there was actually in 2018 a large research study conducted on Salicornia europaea, which is within the genus Salicornia, the one that I'm working with, looking at the root and shoot fungal and bacterial microbiota. And there was some interesting sort of relationships between how diverse these populations were between restored sites and unrestored sites. And when we think about all of the factors that are present in an unrestored site, you have heavier anthropogenic influence, perhaps you have industry or trade happening in these coastal areas, and that can bring any sort of variety of spores into that environment. So there were some really, really interesting trends looking at whether or not those populations in a restored site would sort of level off and just by host plant selection, or if that diversity would remain the same or constant. So lots of really cool research coming out. So for this project, we decided to choose pickleweed for a number of reasons that I will go into in a little bit. But essentially, it's a widespread plant. You'll find it pretty much everywhere along the San Francisco Bay regional shoreline. It's a halophyte, and so it is actually considered to be a model organism for understanding salt tolerance in plants, which, of course, as we know, with overuse of soil and factors such as this, understanding how plants can and do tolerate salt and increases of salt in their environment is going to play an important factor. So we felt that, okay, this plant sort of checks those boxes. And it also provides habitat to some endangered endemic species, including the salt marsh harvest mouse, which some people may be a fan of. They're quite sweet. So for the project, in essence, we go out, we collect our samples all along the shoreline areas. And for our preliminary analysis, just to sort of investigate, are these endophytes present in this plant? Because we have no reference documentation. We decided to go with our first site, which was at the Hayward Regional Shoreline, Cogswell Marsh, which is a restored site. It was restored in 1980. It's about 200 acres of restored commercial salt marsh. And it is a thriving ecosystem. So, you know, we would go out there and just collect those photosynthetic tissues that you see in the images here. All right, so for questions, our questions were, does Salicornia harbor any endophytes? Because we don't have any documentation about that. Is there any evidence of horizontal or vertical transmission if they are present? And do those communities change in relation to the restoration age of the site? And so, you know, in relation to these questions, we believe that based on other, you know, data from these ecosystems, it was likely that there were endophytes present, and that because of, you know, the habitat, they may be considered part of that class 2 that I mentioned a few slides back. So they may actually be transmitted via vertical and horizontal transmission. So we're going to evaluate those in this study. And then we hypothesize that the beta diversity, so the diversity across sites, will differ between sites of differing restoration age. And, you know, that sort of works off of that assumption that a site which is not restored has no sort of regulation about, you know, what type of traffic goes through it or what that environment would be exposed to. And so perhaps there would be more opportunities for a greater diversity of endophytic fungal spores to interact with these plants. So I'm going to talk a little bit about the design and how we sort of mapped out our preliminary data. For my master's level work, we've chosen 3 separate sites relating back to that restoration age. So we have Cogswell Marsh, which we did all of our preliminary data in. We have Giant Marsh over in Point Pinole, which is an unrestored site. And then we have China Camp State Park, which is a protected area. So Cogswell Marsh, we actually were able to compile some GIS data to sort of identify hotspots of where Salicornia pacifica had been documented, even though it was much more widespread than what this diagram will indicate. Cogswell Marsh is glorious. If you have any, you know, it's a little gem inside of the Hayward shoreline. If you go there, it's lovely. You'll find lots and lots of critters running around. It has been, you know, restored, and there were other restoration projects going on not too recently. So we sort of considered it an active restoration site. All right. So Giant Marsh, on the other hand, is an unprotected site. There is a trail that is located at this marsh, and lots of people can sort of go in and out. And yeah, so this site was chosen because it basically had been proposed to be a site for restoration, but it has not yet begun that process. So we were able to secure that permit, and then lots of things changed in the world, but hopefully soon we'll be able to get out there and take a look at those populations of Salicornia. And so those yellow dots I meant, or those green dots rather, that I have on my map here, those are actually documented sightings of Salicornia. But we can, based off of our experience at Hayward Shoreline, we can surmise that there will be large swaths of Salicornia pacifica along Giant Marsh. And for our third site, we have not yet secured the permit for this site, but it is protected land. So China Camp historically was home to a group of Chinese immigrants, I think in the 1880s, I believe, and basically has been protected ever since people stopped living on that land. And there is access, you know, commercial access, but there are portions of the park that are protected. And so these are going to be the coastal sort of areas that we're going to look at for sampling. And this will be sort of our idea of a reference population. So something to compare against. And so we can create a sort of trend of, like, as restoration age increases, you know, how do these diversity levels of these endophytes change, if they change at all? So in essence, we go and collect these little sections of that plant. There's lots of protocols for plating leaf, you know, tissues, which is like involves like a good old hole punch, and you go in there. But with this plant, it was a little bit different. We had to augment some of our protocols just because it is a succulent. But we would collect these portions, plate them out, and then continue subplating until we had what we could assume to be a pure culture. And then once those are isolated, we subject them to PCR analysis, sequencing, and then phylogenetic analysis subsequently. So we'll dive a little bit into that later. But this is sort of allows us to characterize, okay, what do they most closely related to? How closely related are they to, you know, what we have in GenBank, or, you know, we're going to be looking at some other databases as well. So that sort of enables us to get an identification for these fungal OTUs. And so, you know, story time for a moment. When we first did our preliminary sampling, and I was so excited, I had my plates ready, you know, everything ready to go. I put my little segments of pickleweed on petri dishes, and I didn't see anything for a few days. Then about 10 days went by, nothing was growing. I was not too happy about it. And, you know, just decided to put one of those plates under the dissecting scope, and there were some hyphae popping out. So we actually gave it a little more time, and multiple different sort of morphospecies or groups of plates that had a sort of similar physical character were identified. And nearly all segments that were collected of the 10 sampling areas had harboring fungal endophytes. So that was super cool. So we grouped them by morphospecies, which, you know, has its pros and cons. We'll go into that in a little bit. And we actually extracted some of that tissue, subjected it to a quick blast analysis, and we're able to get back some results. And these are the images here of some of the plates which we were actually able to successfully obtain sequences from. And so those DNA sequences were used to sort of fit them into a tree and sort of figure out, okay, what are they most closely related to? How closely related? How strong, you know, how confident can we be in our identification to the genus level? So these were the results for the successfully extracted samples, as well as their locations along the marsh. And so we have Stemphylium pleospora. Pleosporales is really widely represented here. And, you know, it actually was not super surprising what we found once we were able to see that they were there. What was, you know, present on the plate was actually, you know, a group of fungi that are seen to have endophytic associations all around the world. So, and many of them are actually documented to be halophilic. So that was super cool to see as well, albeit maybe a little bit predictable, but that's it. So we used a maximum likelihood model for our phylogenetic analysis. Definitely something I would want to work on, and we will be fleshing these out further. But again, this was just sort of taken from our preliminary data, slapped together. And so I've indicated in the red box here, those are the fungal identifications. So that's actually what we found. We see that they fall right into this Stemphylium pleospora family. So again, these are some of the other OTUs that we blasted and then put into our analysis. So of the majorly represented genera, Stemphylium or Pleospora, Stemphylium is the asexual anamorph. So they're filamentous molds, and that's really how they're characterized. They're very common in soil, and they're often seen as black mold, which is interesting because that is not the pigment that we, you know, viewed on the plate. So that sort of brings up the fact that putting things on a petri dish and putting them in an incubator is, you know, very far removed from its natural habitat and character. And so, you know, it is somewhat challenging to conduct morphological analysis on these tissues. So this Stemphylium, it's a known plant pathogen. It's actually was really, really detrimental to sugar beets in Scandinavia at one point. And that, you know, was really interesting to see that this was living and is living in many plants endophytically, and, you know, with no sort of visible signs of predating on its host. And there is some evidence to suggest that this is a halotolerant species, and this whole group of molds, and they're seen to have the ability to sort of tolerate that salt a little better than one would expect. So we also have Neochimerasporium. Hope I'm not botching these. I just, it's tough to say. They are known to be halophytes. So that was pretty, again, super cool to see. They are not studied in their phylogenetic capacity, so we don't know a ton about how their tree is sort of constructed, and there's constant debate about this particular group. But they are known to be halotolerant, and they are found distributed worldwide in salt marsh, mangrove, and coastal ecosystems. We also have Alternaria that was represented. It's an ASCO. It is widespread, which is not surprising given what we've already found. And then it's also a very common endophyte, so you will see that. But it is, again, another situation where we have an endophytic fungus that lives, as an endophyte in one plant and then can be a plant pathogen or even, an animal or human pathogen. Alternaria is, responsible for causing, fungal sort of allergic reactions in people, and can be somewhat of a detriment to infrastructure and to buildings and things like that, sort of those molds that cause lots of problems for us. But again, just hanging out inside of our Salicornia pacifica all along the coastline. And then we have our last one here. This one's actually really beautiful. I thought she was gorgeous. But again, a widely dispersed group. Important for bioremediation and antibiotic production. This was research that was going on a few years ago, so we'd have to check back in again and see how they're doing with that. But they are commonly found in soils and in halophytes as well, and in the soils of halophytes. So our second phase of sampling, these are some of the plates that we produced from those tissues, and it is on par with what we were able to achieve with our first round of sampling. Which definitely conferred more confidence, ensuring that we weren't just getting, contamination, spores from our environment inside of our samples. We actually were obtaining these from the tissues of the plant. So, we had very, very strong results in our second phase. And so we know now going into our methods, which will be changing in the future, we should be able to get some pretty, consistent results across all sites. And that will lead us into next steps. All right. So culturing pros and cons. We use culturing just to sort of conduct that assessment and to just sort of troubleshoot our protocols. But, really, it's very selective. So not all endophytic fungi or other fungi are able to actually persist in media. It's sort of a crazy metabolic environment to be in, and not something that they would naturally be found in. So we're hoping that our methods going forward are going to yield better results, and we're going to be moving away from culture-dependent methods. So we're going to be using next generation sequencing. We're going to continue with our use of the ITS region for any of you out there who do molecular work. It's very, very familiar. But we're actually going to be using, I believe, ion torrent technology. So that is going to basically open up the floodgates. So anything fungal that can be found in those tissues that can be amplified, will be sequenced. And we'll be able to collect more data, and we'll be able to, conduct more sort of quantitative analyses, including those diversity estimates. So what is the diversity within each marsh? And then how do the diversities compare across each sort of sampling area? Yeah. So that's going to be our exciting next steps that we will be taking on hopefully soon. And then I'll just talk a little bit about the impact. This is not work that produces a lot of really pretty pictures. It's a lot of pictures of molds and plates of molds and plants, which are lovely. But it's a little bit different from some of the other mycology research that may be going on that I might be a little bit jealous about, but not too much. Understanding, in all seriousness, understanding these coastal ecosystems is very important, and really knowing what types of microbiota assist these plants in doing what they do, and exactly what is living within them. Those are both really, really important things to understand that we don't yet understand in entirety. So, these habitats, as we all know, are very crucial, and they are under threat because they are often located near industrial activity, high rates of pollution and runoff, and they're sort of the last stop, right? So everything that runs down is going to flow through this ecosystem, and it provides flood protection, nutrient cycling, all those amazing things. So, sort of shining a light on those specific sort of elements of how they live and how they do what they do. Looking at the microbiota, both in the rhizosphere and in the phylosphere, is definitely something that we should pay some attention to. And, it will help us to understand the habitats that provide food and shelter to some, really, really important species. I mentioned that salt marsh harvest mouse earlier. So, they rely on pickleweed for food, and they rely on it for shelter. And so not only, do we have to be very careful when we go out and sample, we also want to make sure, we're sort of evaluating what is present in that habitat and what this little guy is going to be sort of, in the presence of, right? So, understanding that microbiota is important on multiple levels, and hopefully this research will sort of provide a model for moving into other coastal ecosystems around the world, or perhaps other understudied ecosystems, and perhaps provide a model for evaluating and assessing alpha and beta diversity, and just sort of the diversity of the different types of endophytes that you can find in these very beautiful habitats that are often play a very crucial role in how the planet regulates itself. Yeah. So I just want to take a moment to thank everybody in my lab. It's been, it's been a journey and a half, and we're still trucking. Without the support from MSSF, that we would not be able to do the work that we do. I would not have been able to generate those maps that you saw. So, so much appreciation to, MSSF and to everybody that has sort of supported this research. And hopefully soon we will have some sort of publication that everybody can check out online. Yeah. All right. And that is all that I have for you.
Thank you very much, Jasmine, for this presentation. I will put this live on YouTube, and if you have any comments, you're welcome to share them on YouTube, and I'm sure Jasmine will be happy to give you an answer. Thank you very much again, Jasmine. Sorry for all the technical issues, and we'll see you again for next talk or meeting or in the next foray.
Hope so. Thank you so much. Really appreciate it.
Welcome. Bye-bye.
All right. Bye-bye.